Hello, my name is Andrew Hooker, and today I wanted to talk about model master files or model sharing and how it might be useful in practically implementing model-integrated bioequivalence approaches for regulatory submission. In general, I think there's a great benefit in model sharing. I think it gives an improved reproducibility and validation of scientific results so that other people can take your model and see what it does and actually see what the model is exactly. I typically run a course called Models for Biological Systems at Uppsala University, and one of the course components there is to take a paper and try and implement the model, the population PK model described there. And it's maybe 80% of the time that some components of the model aren't described enough that you can actually reproduce the model and reproduce results in the paper. So sharing models could greatly improve that reproducibility and allow other people to validate your scientific results. It also allows for knowledge propagation, a faster development of new science if you can take the old science, the models that have been developed, and use them for new purposes. Furthermore, it allows for explicit evaluation and verification of the models that have been described or used in previous work without having to extract and assume things that might not be said in publications. So how can model sharing be useful in things like bioequivalence studies? So here is a standard bioequivalence study where we might have a 2x2 crossover design, and we would then have maybe an NCA-based analysis where we would look at AUC calculations, Cmax calculations, and then look at geometric mean ratios and see whether the ratios are within specific limits for equivalence. And there's lots of problems with these types of studies, potentially. You can have things like too sparse of data, so that the NCA analyses don't make sense. You can have other problems with the data that make NCA analyses inaccurate or biased. You might have components of the drug, like long half-lives, which make crossover designs not possible or practical. You might have lots of variability from different potential sources, making it hard to run these types of studies. So there's a number of potential problems with standard bioequivalence designs. So in the last few years, our group has been developing model-integrated bioequivalence methods in order to overcome some of these problems. Using population PK models, we can then look at sparser data. We can handle different levels of variation in a better way. We can handle more of the problems that might occur with NCA-based analyses. The general structure of this bioequivalence method takes some bioequivalence data that's been run and a set of predefined models. And those models have been updated from say, a reference model of the reference substance to incorporate allow you to identify differences in the absorption characteristics between test and reference product. Once the modeling has been completed, then we use our models to evaluate whether there's bioequivalence or not. So our models are then used to simulate what the expected AUCs and Cmaxes and other PK characteristics are in order to evaluate whether there is bioequivalence or not. Inherent in that is looking at uncertainty estimation from our models. So we have to take our model parameters and use the model parameters to simulate from uncertainty distributions in order to draw conclusions about our bioequivalence methods. These methods have been shown for sparse data examples to control overall type 1 error, as well as have a higher power than standard NCA-based methods. That's especially when we have high variation in the data and sparser data. The methods have also been shown to allow for a reduction in study duration. here we have an example of long-acting injectable products, which might use a crossover steady state study in order to assess bioequivalence. In a crossover steady state study, then you have some reference product, which you then look at a steady state PK profile. You switch to a test product, and then you wait until you come up to steady state, and then you assess the PK profile at steady state and compare. With model-integrated approaches, one can look at PK profiles in this test situation before reaching steady state, and then subtract what you have of the reference product in that profile. So research shows that this approach controls type 1 error, but will require more individuals compared to the crossover steady state study, but it can be much shorter. For more information, you can go to this website where you'll see a presentation that describes in more detail what's going on there. So practically, what's needed for model-integrated bioequivalence analysis? Well, what we need is a model or a set of models. So you may have previous models based on a reference product, or you may potentially need to build a model in some predefined way. Currently, we're working on automated PK model building processes. And then you have to adjust those models by adding in effects, differences in the absorption characteristics across products, so that you can assess whether those differences mean that the products aren't equivalent. Furthermore, the models, in order to be able to do this process, need to be identifiable given the study design data, the data that you expect in a bioequivalence trial. Lastly, the models should then be qualified. You should be able to predict what the AUCs and Cmaxs are for, say, the reference product. We advocate using posterior predictive checks for that. So if we think about what's needed then for long-acting injectables, if you look at the long-acting injectable products that have been approved, roughly 33, 17 of those have models in the literature, 27 total models of these 17 products. And many of those models are based on multiple different studies in drug development. So you have lots of individuals, lots of different designs. So components of those models are likely not identifiable in a simple bioequivalence study. So what's needed to be done is then you add in these extra components that you need in the absorption phase to identify bioequivalence or not. You may need to then reduce the model in order to make the model identifiable, and then show that the model can predict reference data in terms of Cmax and AUC calculations. So that process, it would be very useful to have a repository that could store that information and take those models that have passed, that can be used for bioequivalence, so that other people can use them as well. So if we think about model repositories, I was involved in a EU consortium called DD-MORE, which developed a model repository. There's some links here if you want to check them out. In that model repository, there's currently 151 models, ranging from PK models to PBPK types of models. It's publicly available, free to use, searchable. It's supported by peer review, so it allows qualification of the models, although it's only qualification relative to a published article about that model. And when this repository was active, then there was support from conferences like the PAGE conference and publications that was encouraging or even requiring submission of models that were used to be uploaded into this model repository. It's not being used anymore. The last time a model was updated was about a year ago on the DD-MORE model repository. So what can be learned from that experience? Well, I think one thing is that the qualification should be for specific purposes. You could have qualification based on what was presented in a paper or a submission, but you could also have qualification so that you could use this model, for example, for bioequivalence, model-based bioequivalence. You could have different levels of qualification, scientific panel approval or FDA approval. You could have, there's also problems with model repositories. So with many models, oftentimes the data or data structure is important in how the model is described. So you need to share some of the data structure anyway, in order to understand how a model works. This is especially true in non-mem. Clearly it's investment, so you have to have a repository up and running, and you have to pay for that. And then if you want qualification, you have to pay for that as well. I think we're going to talk later in this group, in this set of presentations about IP issues. And of course, people may have developed their models in different modeling languages. They'll have different file formats, maybe different types of data. So I think that model sharing can increase the quality of scientific work. And for model-integrated bioequivalence analysis, a way to share and qualify models for specific products and specific situations would really aid in the practical implementation of the approach and make it sort of more equal for everyone. And I'm happy to discuss in the discussion later on. Thanks very much.